And our next speaker is going to be Ben Garcia.
Oh, you have Dale.
And Ben is going to talk about next generation quantitative proteomics, tools for understanding epigenetic mechanisms.
Thanks. Okay, so thank you very much, Arnie, for the invitation to come share a little bit about the research that's being done in my group for the last couple years since I started my independent position at Princeton University. And as a great lead-in to what I'm doing, we heard just some wonderful research and work coming out of Wade's lab and kind of demonstrating the power that mass spectrometry-based proteomics can bring into analyzing various aspects of biological systems. So I don't think I'm going to have to convince you very much that mass spectrometry is a powerful approach. However, what I'm hoping to do in this time is to show that new proteomic tools that we've been developing in the lab might be nice complementary or alternative approaches to looking at epigenetic mechanisms. Specifically, my lab deals with analyzing histone post-translational modifications. So if you take the amount of DNA that's present in all of our cells, there's meter amounts of DNA that have to be packaged into a very small area in the nucleus. And the way that nature has evolved to accomplish this role is by packaging this DNA, complexing it with histone proteins to form the nucleosome, the basic repeating unit of chromatin, kind of shown here in this microscopy image, this beads on a string kind of image. And this ball of this nucleosome, the basic repeating unit, is made up of histone proteins, 4 core histone proteins, which I'll talk about a little bit more in detail in a minute. Adjacent nucleosomes are attached by DNA, a small piece of linker DNA, and also another histone, also referred to as a linker histone H1. And this nucleosomes keep folding and compacting into the chromatin fiber, and then hence our chromosomes. However, as you can imagine, there are definitely different processes that require both the folding and unfolding of this chromatin structure. So what you're kind of seeing here is the nucleosome and how it relates to kind of a little bit more cartoon image of a higher order chromatin structure, and this is one histone protein, histone H3. So as you can imagine, during various times, during transcription or replication or DNA damage events, this highly compacted nucleosomal structure, chromatin structure, has to be unfolded to allow access to the underlying DNA, to various proteins and transcription factors and proteins involved in DNA damage and repair. Part of this process includes the changes in histone post-translational modifications. For a long time, these histone proteins were thought to be structural static components of chromatin, kind of just there to give rise to this structural aspects of chromatin. However, people started realizing that these histones were highly post-translationally modified. So here's histone H3 with all the different post-translational modifications, kind of a summary of all the different modifications that have been identified to date by various techniques on these histones. And the really interesting thing about these modifications is that several of them are linked to very specific transcriptional states. Histone acetylation in general is linked to transcriptional activation. Histone methylation, which could come as different degrees of methylation, is linked to both transcriptional activation or silencing. It actually depends on the residue. Histone lysine 4 methylation is a mark found on active genes, the promoter regions of actively transcribed genes. whereas K9 methylation, found on the same histone just a few amino acids down, is actually a mark found in heterochromatin, or the silenced portions of the genome. And for a while, it was a little bit unclear of how these modifications were potentially influencing chromatin structure and gene expression. The first model, which is a very simplistic model, thought that maybe these modifications are influencing how the histone proteins are interacting with the DNA. However, when the crystal structure came out of this, of the nucleosome, what researchers found is that the N-terminal tails of the histone molecules, kind of shown here for some of the histones, actually do not complex with the DNA at all. They actually stick out of the surface of the nucleosome. And this is where the most of the modifications, I think the first 50 amino acids or so are the N-terminal modifications that stick out here. Most of the modifications aren't even affecting the way that the histones bind to the DNA. So this kind of surprised the chromatin biologists at the time. And a lot of theories and hypotheses were put forward to try to explain how these histone modifications can influence transcriptional states, and one of them, called histone code hypothesis, postulated by Thomas Genuine and David Alice several years back, says that maybe these histone modifications on these N-terminal tails of the histones that stick out of the surface might act as a binding platform, recruiting proteins leading to transcriptional activation or silencing. And in support, several proteins that have specialized domains that bind specific modifications like methylation and acetylation on histones have been discovered, lending support to this hypothesis. These histone modifications are one part of this new field called epigenetics that has been used, this term recently by many researchers. Epigenetics refers to these heritable phenotypes and gene expression patterns that occur without any type of change to the underlying DNA sequence. And histone modifications are some of these potential type of mechanisms that control this, along with DNA methylation and microRNAs. And if you think about it, all of our starting cells types basically have the same genome, but they have to differentiate into different cell types, tissue types, organs, and ultimately organisms. And epigenetics, might in part allow this progression to occur. Recently, there's been a lot of talk about epigenetics, both in disease and even in the mainstream types of magazines. Here's an example of kind of a trans epigenetic type of effect. These mice are genetically identical, as were their parents. The mothers were fed different diets, one very rich in folic acids, the other one highly deprived. One of these mice, and then when they were raised in identical environments, one of these mice is healthy, as you can see, normal size, whereas both the histone modification patterns and DNA methylation patterns surrounding genes responsible for fur color and also linked to obesity have been altered in this mouse, the one that was from the mother that was deprived during pregnancy and during the weaning process. Epigenetics has been linked to various different other disorders, most particular cancer, and I'm going to talk a little bit about some of the work we're starting to get into in collaboration with some cancer biologists to try to probe histone modification alterations during cancer pathogenesis. So the traditional approach to looking at histone modifications is by the use of site-specific antibodies, kind of shown here. So the first major part of the talk is going to focus on kind of technology development, and then we'll get into some specific preliminary data with some cancer cell types. Site-specific antibodies can be pretty powerful approaches. However, there's definitely some issues that can occur with these antibodies. First is cross-reactivity. So you can see that's actually identifying or detecting modifications on other proteins that aren't the intended target. The other big problem with site-specific antibodies is the phenomenon known as epitope occlusion. And basically what this is if you have a histone modification, so in this case we have a serine 10 phosphorylation, and this is just a peptide ELISA assay with synthetic peptides that all have the serine 10 phosphorylation. So this antibody should recognize all of these peptides. But two of these peptides have lysine, acetylation, and methylation adjacent to the phosphorylation. And this is actually K9 and S10 phosphorylation modifications, which occur in vivo. And as you can see, this antibody recognizes serine 10 phosphorylation quite well, But as you introduce these two modifications adjacent or in the area of what the epitope of what this antibody recognizes, the ability of this antibody to recognize these marks is vastly decreased. So because of this, we kind of favor a mass spectrometry-based approach. We feel it's a little bit more unbiased approach in that we have no issues recognizing multiply modified peptides and sequencing them and determining their local locations, as well as we are developing quantitative proteomic approaches to be able to take state A, state B, healthy disease, or any other types of biological conditions and determine when the histone modification patterns are changing, such as shown here in this just a quick example of low metastatic breast cancer and high metastatic breast cancer cell types. And I'll talk a little bit about this labeling approaches that we've been developing. And here you can see that these two peptides are the same, just labeled slightly different, so that we can tell the differences in the mass spec of which sample is coming from which signals are coming from which sample. And from here we see a decrease in the kind of the higher metastatic, and once we sequence it, we know exactly the modification involved. We use several different types of mass spectrometry-based proteomic approaches in my group. Standard approach is to take your protein or mix your proteins, digest them all up into very small tryptic peptides, and to analyze them by liquid chromatography mass spectrometry, and what is kind of termed a bottom-up approach. And this is a highly sensitive and very accurate and very quick type of mass spectrometry analysis that kind of gives us information on the single modification site at a time. So it's kind of our version of a really quick quantitative Western blot. Whereas, however, I'm going to make the case that maybe we want to look at bigger regions of the histone proteins, the N-terminal tails that have multiple modifications, because maybe the phenotype or the progression of a certain type of gene expression pattern is not the result of a single modification, which is what we can detect with this really, but maybe it's kind of derived from the combination of histone modifications that can occur on the large stretch of the tails. And for this we use a different mass spectrometry approach where we either do not digest the proteins, look at them intact, or look at very large pieces with a slightly different type of approach to get at the same type of information, and that is protein sequencing. So the common way to analyze two different types of samples for expression differences in the proteomes or post-translational modifications is termed SILAC, and that's stable isotope labeling of amino acids in cell culture. You have your one cell culture growing in normal cells, in normal media. So these have the normal amino acids with the common or the naturally occurring isotopes. And in the second set of samples, which may be the biological perturbation or treatment, these are grown in media that contains isotopically heavy amino acids, usually lysine and arginine, with C13 or the non-natural isotopes. Several doublings are taken, after you wait several doublings to get all the newly synthesized proteins incorporating this isotopically heavy amino acids, extract equal amounts of protein, mix them together, and then analyze them by mass spec. And the signals corresponding to the same protein will be easily differentiated because you'll know exactly how many lysines and how many isotopically heavy amino acids you have in the peptide, and you'll know exactly the difference between them. And you can use this for relative quantification. However, although this is a very nice approach to quantifying proteins on a kind of a large scale, a couple drawbacks of this approach exist. One is that there are definitely some cell types, mostly those from tissues and fluids, that can't be easily metabolically labeled. Second, this is a kind of an expensive experiment. These amino acids are not very cheap. And third, especially when we collaborate with a lot of other biologists, they either have never heard of this approach or are willing to attempt it. So because of this, we have decided to kind of make a poor man's working version of this isotopic labeling approach that is based on a post-harvest type of chemical labeling to essentially be able to label multiple samples together. And right now we have created an approach to label 1, 2, or 3 different samples together. And this is based on using a N-terminal propionic anhydride a molecule that labels every peptide. And depending on the molecules and how much either C13 or deuterium they have incorporated, we could analyze 3 different samples and easily tell the differences from these samples. So all these, this is asynchronous growing cells, and then we have these treated with an HDAC inhibitor, butyrate. So we can see the increase of the 3 different signals, and this is corresponding to K14 acetylation, so you get the right kind of data off of this. And essentially we've streamlined, and I'm going to kind of skip over a lot of the technicals, but we've streamlined this approach to be able to quantify basically all the histone H3 and H4 post-translational modifications and the expression levels of most of the other histone variants and some of their modifications in a single experiment. So here's a snapshot of what this data kind of looks like. These are just some of the modifications that we've been able to quantify in a single experiment. This is a collaboration with Catherine Platt at UCLA, looking at cellular reprogramming. So these are embryonic fibroblasts that have been reprogrammed with the 4 transcription factors. You know, these are then basically induced pluripotent stem cells. And in some cases, we see no differences between the 2 peaks. So this was the signal corresponding to the MEF and the reprogrammed cell. But when there are changes, we can easily pick them up, such as here, K18 acetylation goes way up as you reprogram, and you see a decrease of some of the methylation groups. And we can do this experiment in about 2 hours with about a few minutes bioinformatics analysis. And what we're trying to do is then visualize this large amounts of data. So we've been playing with many different ways to visualize, and it's actually coming out much darker than it is on my laptop. But at any rate, you can definitely see pattern changes as we go from partially reprogrammed cells all the way up to these are embryonic stem cells, fully reprogrammed, and these partially reprogrammed cells. And as you can see, there's a kind of a gradient, or there's definitely a kind of trends towards certain histone modifications being important or changing in this process, the significance of which we're still studying. But in general, histone acetylation, especially histone H4 acetylation, you can see some of these other bands of histone acetylation are very important for this cellular type of reprogramming. What we're also interested in developing is methods. Right now we have really quick methods to compare state A and state B, like I just showed you. But we're also interested in trying to get a better understanding of how state A is getting to state B. And for this, we're trying to develop methods to look at dynamic histone modification changes over time. And I talked a little bit about this earlier. This is this quantitative SciLAC approach that people have been using, and we do use this in my lab for comparing just the levels of modifications or protein expression. In the last couple of years, people have been then doing an offshoot of this SciLAC approach, kind of labeled pulse chase SciLAC, where you take your cells, introduce them into the labeled media. Except this time you don't wait until the proteins are fully labeled. You start taking time points along the way. And what this mimics is the pulse part of a pulse chase experiment. And the data, what you should be seeing in the data is the degradation of the old protein and the synthesis of the new protein. And people have been using this to get at the protein half-lives, or the turnover of proteins. And we've also been using this in the group, but we wanted to develop something that was a little bit more post-translational modification specific to monitor just the turnover of the modifications, or the progression of the modifications. So we started off by looking at histone methylation, and if you kind of take a look at the pathway for how proteins get methylated, all protein methylation comes from S-adenosyl methionine, and the sole donor that gets converted into S-adenosyl methionine is methionine. So we figured that if we can grow the cells in labeled methionine, not arginine and lysine like most other people do, but grow them in labeled methionine, obviously proteins that have methionine will get labeled, but then any protein that gets methylated should also be labeled. And what we're hoping to see is kind of the decrease of the old methyl on the histones, and the increase, or the synthesis, or the expression of the new methylation. And here's what the kind of the raw data looks like. These are different panels over different days for H3, K9 monodi and trimethylation. And this is the old and the new methyl. So all these signals correspond to old histone monomethylation, and you see it over the several days. The methylation is actually quite stable mark. You see the decrease of the old just like you would expect, and you see the increase of the new pretty easily in the monomethyl. And you see the same thing in the di and tri, the decrease of the old, increase of the new. or decrease of the old, and then you have the increases of the new di and trimethylation. But something was really interesting when we started looking at this data is that we actually saw signals in the mass spec that corresponded to a peak, this peptide, so it's the same dimethyl group, except that it has one old methyl label and one new methyl label. In the trimethylated species, we also saw intermediates, or what we call intermediates, that had two old labels and one new label, or one old label and two new labels. And what we ended up figuring out about this type of data is that this methylation here, this was previously a monomethylated species that then got progressively dimethylated during these experiments. And these were a dimethyl that progressed to trimethyl and a monomethyl jumping all the way to trimethyl. And why is this important? Well, the enzyme that does this catalyzes the 0 to 2 methyl group jump is G9A, while it's a completely different enzyme, SETDB1, that does the mono to dimethylation. So with this data, we're actually able to kind of infer enzymatic activity of these histone methyltransferases, and these are all different methyltransferases that also are known to induce these different types of methylation state changes. So with normal methods and what we were doing before, we can kind of see an unmodified going to mono to di to tri. But with this new labeling approach, we can see all these different intermediate species. And by using molecular modeling, we can actually determine the rates of either methylation or demethylation of a lot of these species as well, giving us a much greater picture of the methylation dynamics of histone modifications. In general, we can monitor all the histone methylation in a set of series of experiments that are quite easy to do. Most of the half-lives are quite long. So these histone modification and histone proteins in general are pretty stable. If we look at K9 methylation, which is a silencing mark, here's a histone H3 turnover right here in the middle. So it turns over pretty slow. If we look at the mono, di, and trimethylation, the monomethylation turns over really quick. Dimethylation turns over about the level of the general protein turnover, and then the K9 trimethylation is even slower. And this is a general trend that we see on silencing marks. But if we look at a mark that's a more of activating mark, such as K36 methylation, we actually see that activating marks turn over much faster than the general protein turnover. So here again is the protein turnover for histone H3, and then mono and dimethylation of K36 turn over much, much faster. So we have some pretty good tools to compare states and even get at the dynamics of these histone modifications. But now what we kind of have a question of, these are all the different modifications that are present on the histone proteins. We could identify K4 methylation, which is active mark, K9 methylation, which is a silencing mark, on different peptides. But once we digest this protein up into small peptides, I do not know if this modification was present on the same molecule where this other modification was present. And since these are antagonistic marks, activation and silencing, the question then is, well, are these modifications ever found together on the same histone protein? And for my postdoc, I went to Neil Kelleher's group to try to develop methods to look at the combinatorial types of modifications, and to kind of skip through all of the technicals really. Essentially, we've developed pretty much one of the only methods to be able to do this. If you take histone H3, the N-terminal tail, spray it in the mass spec, you see a distribution of all these different modified states. And it's a very complex mess because there's a ton of positional isomers. Even for the dimethylated form, there's about 10 positional isomers with dimethylation at K4, K9, K14, or combinations of monomethylations and monomethylations. If you try to separate these up by reverse phase HPLC, the common chromatography used in proteomics, you get absolutely no separation. By trial and error, we tried many different types of chromatography, and we found that cation exchange hydrophilic interaction chromatography allowed us to fractionate out this really complex mess into something that's a little bit more usable. This is a two-dimensional separation, which separates out histones based on the acetylation status of the histone. And within one acetylation status, we get a second degree of separation based on the methylation content of the histones as well. So now if you take any one of these peaks, throw it in the mass spectrometer, you see that it's much less complex. We can isolate out one of these peaks, do a different type of fragmentation, and from this one peak, from this one fraction, I know that there's two entire combinatorial histone codes present here with these two different marks. Problem with this approach, though, is that it's an offline. This chromatography is very non-mass spec friendly. The buffers used are not compatible as you use high amounts of salt and other non-volatile components. Since this is an offline HPLC approach on an analytical scale, you need a lot of histone to do the separation. And you've just taken your one sample and turned it into about 30 or 40. So you need really long mass spec acquisition times to even complete one run. So it's by far not ideal. And when I got to Princeton, what I really wanted to do was develop a high throughput approach similar to the looking at the small peptides approach that we've developed, a high throughput quantitative combinatorial mass spectrometry approach to look at histone modification. So, we still isolate out the N-terminal tails just like we did before, but now we've developed a novel nanoflow helic version of this type of chromatography that is compatible with mass spec. We spray it directly in the mass spectrometer. Here's the total ion chromatogram separation. As you can see, it's very similar to what I showed you before. You get these pools of 0, 1, 2, 3, 4, 5 acetyls, and within one of these pools you get different degrees of methylated histones. We can fragment these on the fly using an electron transfer dissociation, which is a novel fragmentation approach that has come out in the last couple of years. And we can sequence these histone proteins quite fast now. How we were able to do this is that we're using the same material that's in these columns that we use for the offline approach. The materials is polycat A material, which is a polyaspartic acid backbone. They're usually highly negatively charged, so the histones bind to them quite strongly, and you normally need high amounts of salt to elute out the histones. But what we figure is maybe we can get a similar type of elution if we just protonate the backbone using a pH gradient. So now we start off from a high organic and high pH and drift down to a low organic and low pH. And what we find is we get the same type of separation by just protonating the backbone of this molecule, the resins molecules. So you still get this weak cation exchange type of separation, no salt. It's completely mass spec friendly, so we use it online. And because we've now shrunk what the column size down from 4.6 millimeters to 50 microns, we've dramatically improved the sensitivity and ionization potential, as well as now we only have to use less than half a microgram of histone compared to 100 micrograms. Here's a separation, a heat map showing the separation of all the different modified forms using the new approach. So you have retention time on one axis, so it's just the chromatography. You have mass to charge on this other axis, it's just all the different modified forms. Intensity axis, and the overriding separation is based on the acetylation status of the histone. So you can almost see these pools of 0, 1, 2, 3, 4, and 5 acetylated histones. And within one pool, you get a second degree of separation based on the methylation content of the histones. This chromatography is so reproducible for the most part that we know where one pool starts and the other one stops, and where one pool stops and the other one begins quite easily. Mass spectrometers with these new fragmentation approaches that are implemented on these instruments, we can easily sequence and determine the combinatorial codes of these different forms coming out. So we see here different combinations of acetylation and methylation. And we still have enough sensitivity, even on something that's very low level, that we can still determine, even though the signal to noise is not as great on this mass spectrum, we still have the combinations present, get the combinations present on these forms. We've developed pretty much novel bioinformatics, not only to be able to identify the forms present in this data, but also quantify the forms. I'm not going to go into this, but bioinformatics approach, as you'll see, is really important to help you mine this large-scale data sets. And what we're able to do now is quantify hundreds of histone-modified forms in a single experiment. What we've identified for just the 3 histone H3 variants is over 1,000 different forms globally in human cells. So now the question kind of that is occurring is, if you have thousands of forms spread throughout the whole genome, which ones are truly important from disease state to healthy, or during stem cell differentiation or any other biological conditions? And we're starting to develop other visual approaches to try to get a better handle on this very large-scale data sets. So real quickly, everything I've showed you so far has been looking at histone modifications, either single histone modification sites or combinatorial modifications from global genome-wide extracted histones. But obviously, kind of holy grail and what we try to work towards is one day being able to pull out a specific gene and reading the histone modification patterns from different conditions of a single gene. But that's actually pretty difficult to do. Unlike just analyzing the DNA, where you have the PCR step to kind of help you and amplify DNA, there's no protein PCR step. So what we isolate out from any type of chromatin immunoprecipitation or IP. Protein-wise, that's all we really have to work with. So while we are continuously trying to improve the analytical side of things to get to analyzing smaller and smaller amounts of samples, we still think about, there's 1,000 different histone forms out there of just one of the histones, H3. Which ones of these forms are truly important? And if you believe in the histone code, that these modifications kind of act as some kind of code on top of the genomic code that recruit proteins that bind these modifications, and there are definitely examples of this that have been published, several new ones every year. WD repeat protein binds K4 methylation, HP1, heterochromatin protein 1 binds K9 methylation, BRD proteins bind acylated histones, and so on and so forth. And these are really specific. HP1 won't bind any of these other methylation sites, and WD won't bind any of these other methylation sites as well. We thought about it, and does it really matter if there's 1,000 different forms, or does it matter more if you have a protein that's actually translating or reading this histone code and turning it into some kind of biological output? So we decided to try to develop some experiments to essentially determine what are these histone code reading proteins actually reading. So what we've done is we've expressed these nucleosome binding proteins or histone code reading proteins with a flag tag. So we generate mononucleosomes. Some of these mononucleosomes are going to have this histone code reading protein present on them. Some are not. We have an antibody very specific for the flag tag, so we can pull out the small pieces of the smaller subset of the genome that have these histone code reading proteins reading their histone codes. And then from there, with a fair amount of scale up, we can then remove all the protein, identify all the proteins that are on these chromatin regions, as well as the exact histone patterns that are on these regions, and then take the DNA and go sequence it using next generation sequencing Illumina Solexa instruments. And what we're hoping to do is then build maps of where all of these histone code reading proteins are at throughout the whole genome, and monitoring them under various dynamic processes to see how their interpretation or translation of these histone codes are changing. So here's a pull-down example of these histone codes. Proof of principle experiments are on these BRD proteins, which are transcriptional activators, and heterochromatin protein 1, which is a protein involved in heterochromatin and gene silencing. Here's an input, flow through, and elution. So you see we get a very nice pull down of our bait protein and the histones, and we usually like to see a one-to-one kind of stoichiometry there. If we're lucky, we can actually pull down, if we optimize a little bit, we can actually pull down non-histone proteins as well. So we've identified a lot of proteins that are known to interact with HP1 as well. And now what we want to do is apply the proteomic approaches I talked about before to these type of chip type of procedures. So what I'm showing you here is a BRD chip. I think it's a BRD4 chip against a control histone chip. And what you see in some modifications, there's no overexpression of the histone modifications from these BRD chips versus the control. In other ones, you see huge overexpression of some of these marks either way. And what we can do very quantitatively then is assess all the different histone modifications and determine exactly what histone codes these proteins are interpreting. So there's 3 family members of the BRD family, 2, 3, and 4. And as you can see, they by far like to bind acetylated histone H4, which we can confirm by some Western blotting with a pretty decent antibody against the tetraacetylated histone H4. There's also some other histone marks that are also increased as well, and most of these are acetylation. So these BRDs, the BRD name stands for bromodomain, which binds acetylated histones and shows that we can easily, the mass spec data, it confirms what people have suggested. And now we're incorporating chip-chip type of experiments to see what genes the BRDs on, or chip sequencing type of experiments, what I'll show in a minute. Here's the mass spec data for HP1, which binds K9 trimethylation. I talked about it a minute. There's also a minute ago. There's also 3 different isoforms of HP1, and as you can see, there's a largest enrichment is for the K9 trimethyl, but we pick up secondary enrichments as well on the H4 K20 trimethyl. So maybe that HP1 might be recognizing that, or it might be that this modification might be just present in the same nucleosome as this modification. But our strategies at least tend to work, and we have enough sensitivity that we can actually apply our combinatorial modification analysis to these chipped amounts of material. Here's a control histone, and here's the BRD chip, and you can see the higher amounts of acetylation on the BRD chip, and this is for histone H3, than on the control. And now we can map these back to the genome using next generation sequencing, Selecta by Illumina, or Illumina by Selecta type of sequencing. Here's just a bird's eye view of HP1 and where it's present on the different chromosomes. And obviously, if you kind of hone in on a chromosome or genes, you kind of see that both the genomics and the proteomics actually make a lot of sense and are very complementary. So we saw BRDs have a high amount of acetylation linked to transcriptional activation. HP1 binds K9 trimethylation linked to gene silencing. Here's BRD23 and HP1 from chromosomes 11 and X. These are just in 293 cells, but these are from females, so this is X inactivated. The overlap of the BRD2 and 3 is quite well, and you see a lot more of it than HP1 and chromosome 11, whereas when you go to the X chromosome, you don't see as much of the BRDs, and you see a lot more amount of HP1. So now what we're doing is building these high-resolution maps, and this is not it, but building high-resolution maps of where these histone code-reading proteins are at throughout the whole genome and hopefully under various conditions. So now linking this back to cancer. There's a clear epigenetic component to cancer in some cancers. Several methyltransferases are known to be overexpressed or translocated in different cancers. So what we've tried to do is gather as many different cancer cell types as we can at Princeton University and just do a global scale survey of the different histone modification patterns that we detect. And here is kind of the proteomic data just visualized as a heat map again for these different cancer cell lines. And if you look at this data, you actually see definitely some hotspots across multiple cancer cell lines, and there's a couple non-cancer cell lines as well that we're looking at. And a few things jump out. K9 dimethylation, K14 acetylation, the combination of that seems to be expressed much higher in many different types of cancers. K18 methylation, this is a novel modification site that we've identified by mass spectrometry, also seems to be increasing. This is going to become important for the last part of my talk. Some forms involving K36 dimethylation specifically seem to be overexpressed, as well as some histone H4 marks, some acetylation and trimethylation. So how do you go about then taking this global scale data and trying to understand the role of epigenetic modifications in the pathogenesis of any type of disease? Well, we were really intrigued by this K36 dimethylation mark because it's an active mark, and this is kind of not what we would expect, as well as this novel K18 methylation mark as well. If you look at, so then we wanted to try to figure out if any of these potential histone modifying enzymes were involved in creating this K36 methylation or K18 methylation mark across different cancers, but you kind of have to hone in on a specific cancer or a specific methyltransferase. There's a ton of these different methyltransferases. A lot of them are implicated in overexpressed in many different types of cancers. And mixed leukemia lineage protein actually has its name from being kind of overexpressed in leukemia. Several of this NSD2 family is also known to be translocated and overexpressed in different cancer cell types, as are many of these other histone methyltransferases. This particular family was interesting to us because the SET2D family contains the different NSD family members as well as SET2. And SET2 in yeast is known to methylate K36, specifically doing all degrees of methylation, mono, di, tri. But it's not very clear what these three family members are doing, and if they actually, there's some evidence linking NSD1 to K36 methylation as well. But the really interesting thing was that when you take a look at these family members, especially NSD2, they have a high association with different types of cancers. NSD2 is part of this SET2 domain family of methyltransferases. It's in a region of the genome in Wolff-Hirschhorn syndromes that is deleted, and low amounts of NSD2 are needed during development. And deleting NSD2 deleting this region where NSD2 is present in this syndrome is linked to the pathogenesis of this disorder, which includes mental retardation and other types of developmental abnormalities. NSD2 is translocated in multiple myelomas. It's actually fused to the IgH protein, and because of the high kind of expression by that promoter, NSD2 is expressed 50 to 100 times higher in multiple myeloma. So for this, we have been collaborating quite extensively, both proteomics, genomics, and as you'll see, other biochemical and biomedical type of research with a really good collaborator of mine, Professor Aura Gozani at Stanford University, who's an expert in histone methyltransferase activity. So what is known about NSD2? Well, not a whole lot, and it's really conflicting. Different reports report that it can actually methylate a lot of different sites on different histones. So the first thing we decided to do, we were interested in this protein NSD2 because of the link to cancer, and because it just had not been truly determined not only what it's methylating, but what is a potential role in cancer. We decided to focus on this protein. And the overriding questions that we're interested in is, what type of activity does NSD2 exhibit in vitro and in vivo? And how does NSD2 regulate chromatin structure? And is there any kind of link between NSD2 and the pathogenesis of myeloma? So here's just some in vitro work with the set domain, the catalytic domain of NSD2, compared to a couple other enzymes. G9A is a K9 dimethyltransferase. Set8 is a K20 monomethyltransferase. As you can see, NSD2 in vitro definitely methylates H3, not H4, as some people have suggested. This is just with the catalytic domain, but once we isolated out the intact NSD2, we see NSD2 as well. The intact isolates mononucleosomes, and the signal up here is corresponding more than likely to histone H3. So for this, we took a kind of a candidate approach to try to determine which sites NSD2 modified. So creating a mutant version of histone H3 where all the lysines have been converted to arginines, and then restoring individual lysines one at a time, we're able to tell that NSD2 methylates K18 and K36, which is really interesting to us because those are two sites that we found overexpressed among several different types of cancers, not just myeloma. Whereas SET2, human, only methylates K36. So our conclusion, though, is that there's definitely not any other type of activity such as been reported previously, at least not this in vitro work, on any other sites besides K18 and K36. So a little bit more work to try to figure out what NSD2 is doing within site-specific antibodies. NSD2, as you see here, seems to be catalyzing mono and dimethylation of K36, but not trimethylation. Mass spectrometry analysis of the same type of in vitro type reactions also confirms this. So what you're seeing here is a double quantitative proteomics looking at samples that have no NSD2 or NSD2 added, and you can see the mono and the dimethylation only in the NSD2 added by the mass spec. This is on in vitro, just a recombinant histone, but we also see this at the nucleosomal level as well. NSD2, the catalytic domain, or NSD2, see a higher amount of K36. And definitely, NSD2 is not doing anything else besides mono or dimethylation of K36. It's kind of shown here. The control SET2 is mostly a K36 trimethyltransferase. So you see that there's or no increase in the trimethyl form of histone H3. NSD2 definitely is a K36 mono and dimethyltransferase in vitro. So, but what about this K18 methylation activity? Does this even exist in vivo? And we definitely know it exists in vivo. We've detected it on many different types of cell types. Here's just an example of sequencing the K18 dimethylation. And what we also find is that NSD2 does methylate the novel K18 methylation site. However, it doesn't do it on just recombinant histone. So this is just recombinant histone and the catalytic NSD2 site. So you see no catalysis of the K18 methylation, only the K36. However, when you move to mononucleosomes generated from 293's HeLa cells, you see that NSD2 does in fact exhibit the K18 methyltransferase activity, both the catalytic domain and the full-length protein. And using kind of these synthetic custom histones where we can synthesize these histones with specific marks. We've, in collaboration on Kingston's lab, some people up there, they've made these histones that have specifically K36 dimethylation. As you see, K36 dimethylation histone alone without the NSD2, you only see the signal for the dimethylation. Once you get this dimethylation alone with the NSD2, then you see the K18 kind of coming in. However, the interesting thing was that if we didn't have K36 dimethylation, we didn't see this K18. So this kind of gives us an idea that there's some kind of hierarchy or patterns, combinatorial type of aspect of K18 methylation and K36. And we've also shown that SET2, the only protein in yeast that methylates K36, also does K18 methylation in yeast. So if we go to in vivo now, we have a lot of in vitro work kind of pointing to K36 and K18 being methylated by NSD2. So here's an overexpression cell line. As you can see, you get a little bit increase definitely of K18 and K36. The mass spectrometry data kind of confirms that. K18, K36 are definitely the higher changing modifications in this type of example. Creating different cell lines where you actually can knock out the overexpression of this NSD2 by just recombination techniques. We show that K36 methylation and 18, but this plot's only showing 36 levels are decreased in these knockout cell lines, NSD2 kind of knockout cell lines, but then it's restored in the other cell type. Using shRNA against NSD2, we also see basically the same type of effect. Knocking down NSD2 knocks down the levels of K36 dimethylation and K18 as well, shown there. Here's a mass spec of these knockout cell lines, and as you can see again, the activity of K18 and K36 is the highest in these cells. So the mass spec also confirms the in vivo work, as well as you can see kind of reduced levels of K18 and K36 in these cell lines. Definitely not K36 trimethyl though. So what is NSD2 doing? Hope it convinced you that it's a K36 mono and dimethyltransferase as well as a K18 mono and dimethyltransferase. And when you have no idea what this protein is doing, then you just go to a microarray experiment to try to give you some kind of clue, or a whole proteomic type of experiment. So we've done a chip-chip experiment against different histone marks. And what I'm hoping to show you here is that K18 methylation and K36 methylation overlap quite well, and this is on active genes. And you can see this because K36 trimethylation is actually a silencing mark. It's not on the promoters like K4 methylation is, but it's on the kind of the coding regions of this actively transcribed genes. A summary of this kind of genomic data basically is that NSD2 target genes have a higher expression. They're definitely on the coding regions of these genes. NSD2 chips compared to K36 or K18 methylation are basically co-occupied. And what we're actually seeing is that NSD2 occupancy in these kind of overexpressed cell lines leads to abnormal placement of K36 and NSD2, which we think results in silencing of several potential oncogenes such as c-Myc, transforming growth factor beta, and some other genes which we're not exactly sure of why that would be. And here's just a kind of example of what you're seeing, these overexpression cell lines and the knockout. You see the decreased levels of NSD2 correlate with decreased levels of K36. This isn't the best, so we're redoing this with ChIP-seq data. But for this, we kind of, it gave us enough inclination that maybe NSD2 is being involved in chromatin remodeling through increasing the K36 methylation at genes that are needed to be silenced, such as those potential oncogenes. So new cell lines were created besides this knockout cell line. that restored kind of the wild type NSD2 activity, or we have these catalytically dead kind of mutants, and here's just a characterization of the histone modification changes from these different type of cell lines. We see the decrease in both the NSD2 and the histone K36 methylation in the knockout cell lines, restored in this wild type conversion, and then this catalytically dead protein, you see the corresponding decrease as well. And now what we are trying to study, and we have some preliminary evidence in the last couple of slides I'll show you, is that NSD2 catalytic activity is needed or reconstitutes proliferation rate, anchorage independent growth, tumor formation in mice, and invasion of bone marrow. Yeah. So, okay, last slide. So what we're seeing here is these different cell types that we've created, injected into mice, into anti-parallel needle area into these mice, and you see these KMS that have the high overexpression of this NSD2. You see these cell lines have a GFP marker. Tumor formation is quite high. In the knockout cell lines, it's pretty much gone. And NSD2, the wild type, where it's restored, we again see tumor formation not as high as in these other cell lines, and in the catalytic domain mutant, it's completely gone as well. And some more data showing, yeah, I'm just going to skip to the chase. But at any rate, as you can see now, starting from the mass spectrometry data, we've kind of been working in collaboration and driving forward this, trying to get a better understanding of NSD2, this histone lysine methyltransferase, in the pathogenesis of myeloma. This is the potential model that we think is happening. And just to summarize, developing a lot of proteomic approaches that we think can answer a lot of questions that are difficult to answer by traditional biology approaches. We're focusing on defining histone methyltransferase activity during development and disease and these histone code reading proteins. And here's just the people that have been involved in a lot of these experiments. Great collaboration with Ora Gozani and the funding. Thank you. Sorry for going over.
Before we break for lunch, maybe we have one question, if there is one, burning one. If not, okay, let me tell you, if you'd like to have lunch, you walk right across the campus and there's a cafeteria.